Hello everyone. Today I'm going to talk about a paper called the CO2 control of the respiratory system. In this paper, the authors talking about a biological phenomenon called the short-term periodicities. And they are trying to model the CO2 controlled respiratory system and to find the relationship between the stability of the system and the STP. The paper is organized in two parts. The first one is modeling the carbon dioxide dynamics in our body. They first proposed a 3 compartments model, then reduced it to the 2 compartments, and finally to only 1 compartment. The second part is the system analysis. So in the 3 compartments model, the first compartment is the alveolar compartment. This is like the lungs part. The other two is the muscle and the other tissue compartment. So in this picture, P is the CO2 partial pressure. You can think of this as a CO2 concentration. And VA dot here is the alveolar ventilation term. PI here is the inspired carbon dioxide partial pressure. And PV here is the partial pressure in the venous blood. So when the blood coming out from the muscle, there's a time delayed TV here. And then it comes back to the lungs. This is the PV star. And similarly, the PA here is the partial pressure in the arterial blood. And there's also a time delayed TA before it coming back to the muscle. Okay, now another term is M. M is the metabolic rate of the muscle compartment or the other tissue compartment. Now, using these 3 compartments, we can actually write 5 equations based on the mass conservation. And the author here is trying to reduce this model. So they are trying to merge the muscle compartment and the other tissue compartment into only 1 compartment. But in this equation, we have 2 unknown variables, metabolic rate and the volume of the new tissues. So in here, we only consider the stable situation. So we will like the right-hand side equals 0 and trying to find the fixed point. And after a lot of simplification, we will have these 3 terms and plug them back into the equation. We will have the expression for M. You can find that the metabolic rate of the combined tissue is equal to the sum of the 2 compartments. And for the volume part, the author used the LaPlace transformation because this is a common way to solve the ODE. In here, the Laplace transformation for PM is the PM bar. The Laplace transformation for the first derivative is the SPM bar. So plug back in everything and do a lot of simplification. We will have the expression for PM bar. And do the same thing for the other tissue compartment. We will have the P bar other tissue and bring them back. In here, we only consider when t is very small. So in Laplace transformation, small t means a very large s. So if s is very large, this term and this term can be omitted. That's why we have this simplification and do a lot of things. We will have the V. Now we know the M and V, we plug them back in. This is the equation about the combined tissue compartment. And bring back the alveolar compartment, we will have the model 2. So in the picture here, you can see the muscle compartment and the other tissue compartment is combined in only 1 tissue compartment. Now, In here, the author wants to further reduce the model to only one compartment. So if we omit the time delayed variables, actually we can get this second order ODE. But now we are trying to find in what condition we can omit the second derivative term. So they first use the dimensionless trick. Then they plug in the real data for human being. They find that this term is much greater than 1, so they omit the second derivative term. And also you can find a lot of things you can omit it and plug back in the tau. Because we only have one compartment, so we can change the subscript to S. Now this is our model 3. In this picture you can see we combine everything into one effective tissue. Now to study the stability of the system, the author assumed that the alveolar ventilation VA dot is linearly related to the PS star. So we can plug in the VA dot and get this equation. You can find this is an ODE with only one variable, PS. But this is a nonlinear ODE. So we are trying to find the fixed point and trying to use a linear model to approximate this nonlinear model at the fixed point. So this is a fixed point, and this is the linearized model. So for the stable analysis of this linear model, the author talked about Hayes conditions. If our model satisfies these 3 conditions, then our system is stable. So we plug in the condition and get this term. In here, SI stands for the stability index, and the numerator is the unstable factor, the denominator is the stable factor. If the stable index is less than 1, then our model is stable. So here is a graph about the relationship between the stability index and the parameters. In this graph, we can see that if the inspired carbon dioxide partial pressure Pi is large, or the effective tissue volume Vs is large, then our model is going to be more stable. For the time delayed TD or the controller gain, if they are large, then our model is more unstable. Now about the limitation of our model. First, we can bring back all the assumptions we have made. These are some biological assumptions, but if any one of them are not satisfied, then our model may be wrong. Another limitation, I think, is that this paper has little simulations. So in the original paper, they said exhaustive simulations is a formidable task. But because this paper is written in 1988, but now in 2022, I think we can do more simulation. The third limitation, I think, is when the author is trying to linearize our nonlinear model. They didn't consider the case when the fixed point is a hyperbolic. So if that is the case, our model may not be applied. Okay, thanks. That's for my presentation.